migrant gradually die, leading to the malfunction of this pathway and the characteristic motor problems. Drugs that replace or mimic dopamine are often used to treat these motor deficits, but they grow less effective over time. Deep brain stimulation may also be used to treat symptoms. However, no current treatment slows the neurodegeneration. A distinctive pathology in most cases of Parkinson's is clumps of misfolded proteins within neurons. Lewy bodies are the most common type. A characteristic component of these is a misfolded protein called alpha-synuclein. These molecules can form small repeated units called oligomers, or longer fibrils. Mounting evidence indicates that these are toxic to neurons and play a key role in driving Parkinson's. Unwanted proteins are normally cleared by the cell's different types of protein-degrading machinery. Some evidence points to these systems being overwhelmed by misfolded alpha-synuclein, and this might be a key factor in what kills the neurons. Parkinson's has also been linked to problems with mitochondria. Mitochondria provide cells with the energy to perform vital functions. They're highly dynamic and can fuse together or break up into smaller versions in response to a cell's energy demands. They can also be transported to areas of a cell that need them the most. However, in Parkinson's, these processes can be impaired, and mitochondria are unable to sustain proper neuronal function. As they become old or damaged, mitochondria are removed and replaced. Again, this recycling is thought to be disrupted in Parkinson's, leading to the accumulation of damaged and worn-out mitochondria. Another idea is that the cells called glia surrounding neurons may play a role in Parkinson's. As dopamine neurons are lost, one particular type of glial cell, called microglia, is thought to take up the resulting cellular debris, triggering an immune response. Once activated, they release inflammatory cytokines, which activate neighboring microglia and another type of glial cell called astrocytes. Chemicals released by activated microglia and astrocytes have been shown to injure neurons. It remains unclear which mechanisms drive the disease process in Parkinson's. What is clear is that with time, more areas of the nervous system develop pathology. And one emerging idea is that this happens through the transmission of misfolded alpha-synuclein. While the multifactorial nature of Parkinson's disease progression might make it hard to fully understand, it does provide a number of potential therapeutic targets for slowing or even halting the neurodegeneration. For example, enhancing the clearance of abnormal proteins and blocking their transmission, improving the function of mitochondria, and targeting the neuroinflammation are all currently active areas of research.
So this video has everything that my research group and what we are researching from past almost 5 to 6 years. So let me take you to the little more into the deeper. This is SNCA gene, which is being transcribed by the fourth chromosome on the Q21 arm of the SNCA gene, which transcribe this particular protein called as alpha-synuclein. So you guys may be knowing that the typical protein synthesis process start from the two checkpoint. One is a transcription and the one is translation. So the exons are getting cleaved off from the introns, and they are making of about the 5 exons, which comprises of the SNCA mRNA, and eventually it is going to translate it into the alpha-synuclein gene, alpha-synuclein protein, which is made up of the NAC regions, carboxylic end, and the amino ends. And then eventually this alpha-synuclein undergoes varieties of post-translational modifications, which is not limited to, but there are varieties of post-translational modifications are there. I'm going to talk in the next slide. And these PTMs eventually makes the alpha-synuclein to get inside the intracellularly to the different organs, and it performs its physiological role in the native form. I'm talking about the native form. So unfortunately, what happened in case of Parkinson's disease, this particular genes undergoes varieties of dysregulations, which happens at the post-transcriptional and the post-translational. So my research group, half of the PhD students and master students works on the miRNA dysregulations, and half of them works on the PTMs. Especially on the PTMs, my research group is focused only on the phosphorylation. So what happened, this particular genes which is getting mutated because of whatever the reason, and also in case of the miRNA dysregulations or PTM dysregulations, the monomeric form, which is the native form, I would say the physiological role, is going to get compromised because of its conversion into the oligomeric form. So oligomeric form is highly toxic in nature, which is not dissolvable. So when it's get into the cytoplasmic area, it is going to affect almost all the important macromolecules of the cells, white, including but not limited to mitochondria, lysophagy, ER stress, and nucleus as well. So my research group is focused only on the mitochondria and the lysophagy. So which is what I'm going to talk about. Few of my research which is focused on only on the mitochondria and the lysophagy. Moving to the next, how does actually post-translational modifications happen at the more deeper way? If you look at the different regions of the, in case of disease state, this particular protein, which has N-terminal, C-terminal, neck regions, all of them have nearly about 140 amino acids, composed of 140 amino acids. All of them are going to went through the varieties of post-translational modification, and these modifications are being driven through the varieties of enzymes. So now these enzymes which drives the PTMs using different kinds of mechanisms makes this monomeric form into the oligomeric form, and this oligomer eventually make the beta sheets or protofibrils, which eventually get accumulated and form the Levy body, and they damage the mitochondria and they eventually cause the neurodegeneration. So I'll be focusing on few of the research which my students has done on the how these oligomeric forms eventually cause the mitochondrial dysfunction, which you guys just now have seen in case of video where the transport of the mitochondria happens through the exons from the enterogate to retrograde transport. So I'm going to give the flavor about the how the mitochondria play essential role in the neuronal survival. So what happened in the next step once the oligomeric form of the alpha synuclein happens, which eventually going to get inside using the VDAC channels into the intracellular and into the intramitochondrial um, transports, which is eventually going to cause the damage to the mitochondrial DNA. It is also going to, disturb the complete fission and fusion process, and also going to cause the disbalance between the pink park independent, ubiquitinase pathways, which eventually going to disturb the ER stress pathway. It is eventually going to cause the disturbance in the mitophagy. And I'm going to talk about few articles which are related to the mitophagy as well. So that's how the oligomeric transitions of the alpha synucleins play a great role in modifying the normal physiology of the mitochondria. Hope the audience are with me.
Yes, sir.
Okay,
Yes.
so as a part of my drug discovery, I'm going to talk about the bioinformatics approach. As I mentioned, that in order to find out the more precise and more validated target, instead of going into the blind drug discovery, which was most of them are followed because of whatever the reason, because of limited resources. I won't say that it is limited to the every institute, but the institute like snipers and national institute, they do have a good amount of facilities. So we are using those facilities in order to find out the very precise target using the particular data which are already available. So we have used the geo data series, which is of belongs to the nearly about 98 Parkinson patients, which are of familiar in nature, and 94 of the control patients. So what we have done the meta-analysis and differential gene expression analysis based on the p-value and the Fisher values. What we could be able to see that there were nearly about 190, 109, 1977 genes were differentially expressed, among which 980 were the upregulated and 997 were the downregulated. So what we got the differentially expressed genes among the comparing with the control in the PD patients. So now we wanted to find out the functional annotations based on the geoterminology. Since we are focusing on the kinases, which drives the post-translation modifications, we gave input of the molecular functions of protein tyrosine kinase, transmembrane receptor protein kinase, and transmembrane receptor protein kinase. So these were the three terms which we have used. Based on those terminologies, when we applied the Panther classification system, we could be able to see that there were nearly about 72 protein kinases were found to be differentially expressed among all these differentially expressed genes, and we could be able to see that there are nearly about 33 kinases were upregulated and 39 were downregulated. So in order to further understand now we have a kinases, and since we have a focus, we are focusing on our particular gene, which is SNCA, which transcribed the alpha synuclein protein. So now we wanted to see the protein-protein interactions between the 72 kinases, which we have narrowed down from our previous approach, and we try to see that what are those kinases which are really um, new other than what is already explored. So we could be able to see that there was CSNKA1 and MEBK1 were the so far not explored in the context of Parkinson disease, having their interaction with the SNCA. So to further narrow down, we have done the, using the Seroninja Bioluminate software, we have done the SNCA plus top four upregulated kinases interactions based on the H bonding, salt bridge, and pH stacking. We could be able to see that there are nearly six, kinases which were not so far explored, and we wanted to see that what happened with these six kinases with the SNCA using the MIR net analysis, where we wanted to find out the post transcriptional analysis of the miRNAs, which is eventually going to potentially modify it during the transcriptional steps. So we could be able to see that there were nearly about 8 miRNAs potentially targeting the SNCA with the at least 6 to 3 kinases, which we have narrowed down from our previous approach. So now to further understand what we have done, we wanted to see what exactly happened with those, potentially 4 kinases, which what we are believing, and we have tried to, explore with the standard available so far. I would say that the kinase called as ABL kinase. When we compare our data with the Piper pose energy, we could be able to see that our four kinases, which is CLK4, CLK1, CDK6, and MKNK2, which were found to be highly interacting with the SNCA gene and having almost the same energy score and Piper energy. So we could be able to see that yes, all these four kinases can be used as a target in order to prevent their interaction with the SNCA gene in order to prevent its oligomeric transition. So the work is right now going on. So what we are doing, we are going to knock down these kinases using the siRNA transfections, and we have seen that there were significant results, which I'm not going to show you right now. We have seen that yes, there was a significant involvement of these kinases are there with respect to modifying the alpha synuclein. There are pretty nice data are there, which I'm not going to show you right now. The other approach that, as I mentioned in my, you could have seen in videos that my group is focused on the neuroinflammation and immunity. So one of the PhD students was trying to find out the HP2X7 receptor, which is highly involved in the Parkinson disease because of this involvement in the varieties of neuroinflammatory pathways. So in which this is called as the hit-based drug discovery, and previously what I showed it was the target-based discovery. So what we have done, we have used the homology modeling of our target receptors, and we selected the library compounds of nearly about 3,521 phytoconstituents. Based on the Lipinski rule, we could be able to filter, narrow down them towards about 2,142 compounds. And we have performed the high throughput virtual screening. We could be able to find out, narrow down nearly about 642 compounds. Using the standard precision docking with our targets, we could be able to narrow down 192 compounds. And then we have done the extra precision dockings, where we could be able to see that 57 compounds were narrowed down. And then we have done the post-docs MMGBCA, and then we could be able to see that there are about 30 compounds which were narrowed down. And then we have done the ADME, which is just now I showed you in case of my one of the previous slide. We use the EDMET and ToxPronet softwares, where we could be able to see that out of 30 compounds, we have narrowed down based on the toxicity profiles and ADME, we narrowed down to the 10 compounds. And from those 10 compounds, we again went for the molecular, simulations, where we narrowed down to the 5 compounds. And these 5 compounds right now are being used for the network pharmacology to again further do the molecular simulations using the system pharmacology software, which we don't have right now. It is CDR I have, and the work is going on. Hope the data will be available soon. And one of my PhD students who focus on the immunity and inflammations, what he has done, he would like to focus on the innate immunity, especially focusing on the toll-like receptors, where he wants to find out which microRNA actually is regulating the toll-like receptors. Again, he has used the geodatabase series, where he used the GeoSeries from the Gene Omnibus database, where he did the sample selection of the patients from the normal populations of about 8 and the 12 PD patients, where he did again the GOR analysis, and we could able to see that differentially expressed genes which are about 18,000, and then he did the heat map analysis and the volcano plot based on the highly enriched gene analysis and assessment based on the innate immunity. He could be able to find out the narrow down to the 10 top hub genes, and then he did the MIR network analysis, which MIR is actually interacting with the top 10 hub genes. So I'll show you the few data. So this differentially expressed innate immunity in the patient sample shows that the different fold change of the log FC value is 2. If you could see that the red one is the highly expressed upregulated one, and the blue one is the downregulated one, while the black one is non-significant, regulated or dysregulated genes. So then further he focused on to find out those biological process which belongs to those particular genes. Again, he was focusing on immune response, and then he narrowed down his construct by the Synogy and Panther software. He find out that 5 categories of the immunity system, which is again immunity. Then he narrowed down to the innate immunity, virus interaction, apoptosis, and adaptive immunity. So he could be able to see that these are the majorly ones differentially expressed immune pathways involved in those genes. And the next step, he see that the heat map analysis between the control patients and the PD patient, he could be able to see that the differentially expressed gene which belongs to the innate immunity were highly expressed in case of Parkinson's disease. Finally, he wants to see that what was the enrichment that is based on the especially toll-like receptor signaling, which was found to be highly significant among all the pathways which he was seeing in case of fold enrichment. And then further he did the heat map analysis of only the toll-like receptor. He could be able to see that the PD patient has shown to be varieties of toll-like receptors were highly expressed in case of PD patients as comparing to the normal control populations. Further, he wants to see that how these toll-like receptors, which we have seen just now in those TLR4, 7, 6, and all those three, he wants to see that how they are going to be associated with the neuroinflammatory pathway, where he used the CAG pathway, which we have is available softwares with the TLR signaling. He could be able to see that there are varieties of pathways are there, but his focus was on JAK-STAT pathway. And then he was trying to find out what is the PPI network analysis of the innate immunity based genes which are found in the PD patients and the normal control patients. And he tried to see that the innate immunity differentially expressed in the PD patient with the confidence level of 0.4. And he could be able to say there are nearly about 314 are the most nodes which you could be able to see in the expected number of ages are about 707. When he did the PPI network analysis of the only toll-like receptor genes, he could be able to see that those TLRs, especially what he had seen in case of previous slides, he was focused only on the TLRs. He wants to find out which were the top 10 hub genes which are going to have interaction with the toll-like receptor signaling. Then he used the software called the cytoscape software. He could be able to see that there is a great amount of red color genes who are fusion events, and blue are from the genes of co-occurrence genes. So further he wants to find out the miRNA network analysis. So based on those TLRs and the miRNAs, he could be able to see that based on the miRNA database, he could see that there are nearly about 5 miRNAs which are regulating the TLR pathways. So now the next approach is to find out whether these 5 miRNAs are really involved in the PD or not. So he is again trying to do generating the stable cell lines of knockout based, and then he would see that whether the knocking out of those genes are really regulating these immune, especially toll-like receptor signaling or not. Finally, he did the identifications of the pathological conditions with the miRNA, which he identified based on the different disease conditions, where he could be able to see that, yes, all these, miRNAs are having a great regulations on the cerebral ischemia, immunoregulatory role in the peripheral and both the central immune system, which is what his focus on, because we don't know whether the immune systems comes first or whether the alpha-synuclein comes first as a part of the pathogenesis of Parkinson's disease. There have been plenty of reports that yes, there are varieties of miRNA there which are involved in the AB deposition, the Alzheimer's disease as well. There are few miRNAs which are already reported. They are highly express in case of breast cancer as well, which is being used as their biomarker as well. As I mentioned in one of my videos, that microtubule is what they play important role in moving the mitochondria from the enterogate to the retrograde transport, since the dopaminergic neurons are highly demanding in the energy because they are highly dynamic in nature. So the enterograde and retrograde transport, the mitochondria across the exons is highly regulated by the varieties of adapter proteins, which is one of my students has recently done this work to find out, because of the alpha synuclein accumulation into the dopaminergic neurons, how does it actually affecting the mitochondria, its autophagosome formation and lysophagy on the mitochondrial tubule acetylations. And then few of the students just works on the mitophagy to find out how the Parkinson disease can be enhanced using the dimethyl fumarates. We also done the dimethyl fumarates effects on to find out the NRF-TIG or LEMP cathepsin pathway to understand the autophagy pathway. And we also done the study on fingolimod, which is one of the drug candidate approved FDA for the, I think, cognition. So it is going to regulate varieties of the lipidomics profile in case of the brain. So where we have done the BNIP pink dependent mitophagy pathway, where it explores effects at the neuroprotective. And we also use the fluoridine for its autophagy pathway, especially on mTOR and NRF2 P62 mediated in case of mice model. So I'll give you flavor of the one of the article what we have done recently in case of microtubule mediated exon commute, which you could be able to see that the mitochondria movement across the exons during the energy demands, which is being fulfilled by the mitochondria, healthy mitochondria, it is regulated by the retrograde and anterograde transport. It is believed that the retrograde and anterograde transports are getting affected during the Parkinson disease because of damage of the mitochondria, because of the mitochondrial inclusion of the oligomeric alpha synuclein, and also it is going to affect the adapter protein, which are nothing but the dynein, dynactin, and kinesin. So the student has made the hypothesis that, sir, I wanted to see whether what happened to these adapter proteins with respect to the mitochondrial movement across the exons, where she wants to use the particular drug called as biclin, and she wants to prove by using the Parkinson models. So she has used the rotenone in the ICV, which is intracerebroventricular injections, and the surgery, where she's done the exclusion inclusion criteria. Based on the exclusion inclusion criteria, she included those animals which are compromised based on the behavioral test, and then the animals subjected for the drug treatment of biclin through the oral routes for about again 28 days. And then on the end of the day, we again performed those behaviors, and then we did the biochemicals, molecular pathways, to understand what happens to those adapter proteins. So she has done the first assessment of the animals, whether they are showing the oops, the videos are not there. I wish if I could be able to show you videos. It's not working. Fine. So this is called as the ipsilateral and contralateral movement, where we wanted to first see that yes, the animals are showing the compromise into the intact sites and the damaged sites, and then those animals which are shown to be compromised in their behaviors, we eventually take them for the main studies, and we could be able to see that on the 0 day animals were able to remain on the revolving rod for almost 300 seconds, but on the 21 days or 28 days we could be able to see that rotenone used animals was severely compromised, and with respect to the drug treatment they were able to restore their behavioral patterns. And then we did for the exploratory behavior, especially with the open fields, where we could able to see that animal with the rotenone controlled treatments were shown to be significantly reduced their exploratory behavior. However, with the drug treatment it was significantly improved. And to assess the Parkinson marker, especially the TH and alpha synuclein, we could be able to see that the rotenone could be able to significantly decrease the tyrosine hydroxylase, which is a rate limiting enzyme, which is responsible for the conversion of the L-dopa to dopamine, and alpha synuclein, which is the pathological hallmark, is significantly expressed, which was got normalized with the two different dose levels. That shows that yes, our drug could be able to reverse the phenomena of the Parkinson disease, and we wanted to see at the very molecular level in order to confirm with the immunofluorescence. We could be able to see that the rotenone could be able to damage the substantia nigra greatly. However, with the drug treatment could be able to restore the or could be able to provide the neuroprotective mechanisms on the dopaminergic neurons when we compare the Th positive cells. And finally, we wanted to see the autophagy markers with respect to LAMP and P62 and LC3, which was significantly got compromised. LAMP1 and LC3 in case of the rotenone, which was got significantly improved with the drug treatment, and P62, which was got downregulated, upregulated in case of the rotenone treatment, which was got normalized with the Biclin of two drug treatments. Finally, we wanted to see what happens to those adaptor proteins which are responsible for the microtubule associated motor proteins for the movement of the mitochondria across the membrane. We could be able to see that the dynein, dynactins, and KIF, all of them got compromised, which are responsible for or which are adapter proteins which helps the mitochondria to move from the retrograde to anterograde transport got compromised. However, uh, there were no significant results were found between the alpha and beta tubulins, which was very surprising to us. We don't know what has happened to this because the student has left, so we don't have anybody to to look after the reason behind the no significant change in the alpha beta tubulins. So, what conclusion we could be able to make that the during the Parkinson disease, the exon transport get compromised because of having said that effect of the oligomeric alpha synuclein, which damage the mitochondria, it also going to damage the adapter proteins as well, which was improved via the autophagy mechanisms, and the retrograde anterograde transport could be improved. However, the only lacking, I would say, the drawback of the study is that we wish, because we already designed, but because of the time limitation, student has to go out for our higher studies. We couldn't be able to do the live cell imaging. We wish if we could be able to complete then, and that could have provide us the more confidence about these effects on improving the motor protein expressions. This is another study. I think I'm limiting with the time. I'm not going to show the mitophagy data of using the immunofluorescence, again the behaviors, protein data, immunostaining. We have a varieties of PCR data also. Finally, I would like to conclude that the research is not limited to the any specific tools or techniques. There are varieties of approaches are there where you can serve the suffering society via any means. It is not limited that you are supposed to do the only genomics, only proteomics, or you have to do bioinformatics. You have to do cell culture. You have to do the animal studies. But there are varieties of other thought process that may exist into the world to serve the suffering society. I'm going to show you a few of the examples that I came across, and my God, this video is not okay. Good, it's working. This is the magnetic button, which is one of the challenge for the Parkinson patient, which they have a the shaking movement, uncontrolled motor movement, so they couldn't be able to perform the fine motor movement actions. So this is just a small innovations, but having a great impact on the patients who are suffering from Parkinson disease who couldn't be able to tie up their buttons. The other example I would like to show here is called as anti-gravitational, anti-shaking spoon, which is again a difficulty for the patients to eat. So I have seen this spoon personally in one of the international conference somewhere in the France. It's a very innovative idea to serve the suffering society. That is what I want to convey, that the research is not always limited to, but the ideas can be anything to serve your the field whatever you are in. Thank you everyone.